Hello, friends. I don't know about you guys, but I am so glad to be back from lunch. My stomach is feeling great, my brain is feeling good too, and I'm really excited to speak with you guys today about digital biology, the life sciences, and biotechnology, and how life as we know it today is going to be drastically different in the future, that biology is the technology, and that you, every single one of you in this room, is positioned to change the course of life on this planet and on other planets too. So no big deal, right? You guys just had lunch. You're ready to talk about the future of everything. So whenever people talk to me about life sciences, they always say something like, you know, my company doesn't do anything with healthcare. We don't do anything with agriculture. The life sciences just doesn't have anything to do with me. And I know as a biologist that I'm, of course, biased, but what I'm hoping to convince you of today is that every single one of you is being directly impacted by biotechnology in the life sciences. And that's only going to accelerate in the future as these technologies that I'm going to introduce you to today start touching every single one of our lives. And we're going to be talking about the 4 big categories that I've highlighted here on this slide, which are agriculture, healthcare, species or the environment, and then let's think a bit about innovation, making, and what the future is going to look like. If you eat food, if you have a body, if you live anywhere, you should really care about the life sciences. So the other day I was talking with a colleague of mine at Singularity named Chris Cowart, and he put this really powerful metaphor in front of me. He said, you know, as we're talking about these technologies, we are raising really important questions. And you've heard my colleagues today, Nathaniel, Anita, everyone's been raising really hard questions for you guys. And I'm going to continue to do that today. I have a lot more questions than I have answers. But here's what I want to ask you to think about as we're taking this journey together. So think about a road, right? Technology is accelerating and it's leading us down this path, and the future is starting to look like a pretty scary place. And if you think of the future as an end, like a cul-de-sac or a dead end, then there's only one way out, right? And that's to go backward. And I'm going to propose to you that going backward is actually not even an option for us, right? So keep in mind today this idea, just because we have these big questions about technology, questions that we have to answer today, not our kids, not our grandkids, ourselves, we have to be designers and makers and figure out how to turn a dead end into a roundabout. So let's talk about biology, let's talk about digital biology, and I'll explain the digital piece in a minute. But just as a quick review of kindergarten biology, all life on Earth is related. All life on Earth is made of cells, including yourself. And if you zoom into a cell and you zoom in further and further, eventually you get down to the level of the chromosome, which is essentially like the manual that has your source code and your operating instructions in it. And if you zoom down even further into the chromosome, eventually you get down to the level of the famous double helix that Watson and Crick told us about the structure of back in the 50s. And that double helix is made up of 4 letters or 4 chemicals that for short we call A, C, T, and G. Now here's where the digital part comes in. A, C, T, and G is how life stores information the way that computers store information in zeros and ones. It's the source code of life, and all life on Earth runs on the same operating system, which is DNA. And as it turns out, that operating system is also open source. We're going to come back to that idea in a minute. But when we think about our DNA, your human DNA has about 3 billion base pairs in it, and 99.9% of your DNA is identical to that of every other person on the planet. So I'm going to say that one more time because that thought helps me get out of bed in the morning. 99.9% of your DNA is exactly the same as every other person on Earth. That tells me that a lot of the reasons we kill each other, they're not hard-coded, right? We make all that stuff up. I live in California, we talk like this a lot. Um, but for me, what's really exciting about that is this idea that all life is related means that anything that I can do to digitize this information now makes the information of life accessible to these exponential curves that you've been hearing about all day. So how do we digitize DNA, right? When I think about DNA, I don't think of that chemical molecule, the double helix. I see this. I see computer code. And when I think of this as a digital framework, as digital biology, then anything I can do to computer code, I can also do to DNA code. So let's just think about that for a minute. So let's say what I wanted to do was move big chunks of code around. Well, that's called genetic engineering. We've known how to do that since the 1970s. Let's say I wanted to write DNA code from scratch. Well, that's synthetic biology. We're going to talk about that a bit more today. And let's say what I really want to do is go into the code and debug the code one letter at a time. That's called gene editing. It's one of the sexiest things out there. You've probably heard of a technique called CRISPR-Cas9, which is like a pair of programmable molecular scissors that now let scientists precisely and permanently, and a hell of a lot easier than what I was doing in grad school, change DNA code one letter at a time. So that's really, really exciting. And where does all this digital information come from? Well, it comes from the process of taking that chemical molecule of DNA and then turning it into some sort of digital form that we can do operations on the computer. So this, of course, is a singularity talk, so I have to show you an exponential curve. So please note the log scale on the y-axis, friends. Okay, so back in 2001, you probably remember the publication of the human genome, the draft version of the human genome. It took 10 years to complete, $2.7 billion to build, and it was a draft. Like it wasn't that great, all things considered. $2.7 billion will get you a really great fighter plane. So after 2007, what you can see is the price performance of DNA sequencing was approximately following Moore's law until 2008, when a new form of chemistry and analysis for DNA sequencing was invented, and given the unfortunate name of next generation sequencing. We're actually on to the new version. What do you think it's called? Next generation, yeah. I'm not to blame for that, by the way. So what you can see, though, is after 2008, the price performance of DNA sequencing began radically outperforming Moore's law until 2015, when we got to the point where it cost $1,000 to sequence an entire human genome. In the United States, that's cheaper than an X-ray, which is saying a whole lot of other things about our healthcare system. But today, you can get the exome of your DNA sequence, meaning just the bits that we know do stuff, for about $200. It's become incredibly cheap and incredibly fast to do DNA sequencing, which is radically changing how we can think about life as source code. And in 2001, this was the type of room that it took in order to sequence DNA. You had, they're still there, actually. If you go to Boston, you can still see these warehouses, no windows, all filled with these giant machines, which cost millions of dollars a piece and were about the size of a refrigerator. So that was in 2001. This is what a version of a DNA sequencer looks like today. It's handheld. It plugs into a laptop via USB. And this is made by a company called Oxford Nanopore. They're also working on a version of this device called the Smidge Ion, which plugs into your smartphone. And just the other day, in fact, last Friday, Oxford Nanopore launched a new device, which is called the Minute, which is a handheld AI supercomputer that does real-time DNA analysis with an NVIDIA AGX chip, which runs on GPUs. So massive computational power, again, in the size of a handheld device that plugs into a laptop. Which is insane, right? That means you can take a DNA sequencer and put it in your pocket and take it wherever you need this information. So farmers in Africa have already used this system to rapidly diagnose a cassava disease outbreak that was decimating crops here in Africa. But this democratization and dematerialization of this technology has really big implications, right? What does it mean when everybody's walking around with a DNA sequencer in their pocket? I just told you that your DNA is your source code. So what does it mean about DNA privacy? Does that even mean anything? You might have heard some reports recently that if some researchers get a copy of your DNA sequence, just some of it, they don't even need the whole thing, and a Facebook access, they can figure out who you are. Now you might say, okay, Tiffany, well, I'm just gonna be really careful with my DNA and I'm never gonna let my sequence get out anywhere. Well, the problem is you are leaving DNA behind you every second of the day. Everything you touch, your lanyard, your pen, your glasses, the seat that you're sitting on, the cups that you use, every single one of those things you are leaving behind your DNA. And I could wait until the next break when you all get out to leave, and I could go to all of your cups and take your DNA and sequence it with one of the min ions that I have. I'm not going to because I'm very busy. So that's not going to happen. But when you start thinking about the future and all this DNA data, questions like this start arising. What would you do, even when you get your DNA sequenced, what are you supposed to do with it? How do you extract actionable insights from that? And I always encourage people to go play with the technology. And if you go down in the exhibit hall, we actually have a DNA testing company here that you can go down and talk to. And one of the things that I think that they're doing really, really well is being scientifically rigorous about what they can really say about DNA. You know, capitalism is great, but people can sell you anything for anything, right? So remember to use your critical thinking hat whenever you're buying these types of products, and think about, what will I really be able to do when I have this information? But one thing that I think is really important about this democratization of this technology is it means that the places like South Africa or Africa in general that historically have not been sampled for any type of big DNA analysis, right? Like pharmacogenomics, for example. Using your DNA to figure out which medicine you're going to respond to the best, that's almost exclusively run on white Europeans and white Americans of European ancestry. That's not going to help the vast majority of people in the world. So South Africa's genetic diversity is rarely included in these analyses. And for personalized medicine to really make a difference in Africa, I think it's critical that African populations and African genomes are being treated as, of equal value as the types of data we've also always used before. And not only is it scientifically important, it's just right? It's just right to be capturing that diversity. So there are programs like the South African Human Genome Program who are distinctly working on enriching the world's knowledge of the genetic diversity that's contained in Africa. Africa's populations are the oldest populations, which means they have the most genetic diversity in them. So it's a really, really exciting opportunity. And this is really important because DNA and other types of biological analyses are setting the stage for a radical shift in healthcare. Now, you probably know DNA is not the whole story. I've given you a very, very simple version of the story. There's DNA, there's RNA, there's proteins, there's metabolites, there's histones, there's epigenetics, there's all kinds of amazing stuff going on, and we know how to digitize all that information. And we're getting better and better at putting it together in ways that become predictive and that can deliver personalized insights. So what we're seeing is a radical shift away from what healthcare looks like today, which is mostly reactive, right? You wait until you have a headache before you go to the doctor, and the doctor does a brain scan and realizes you have a tumor. That's reactive medicine. That's what we're doing right now. But these types of large biological data sets and really amazing advances in sensors and DNA computing and all these technologies you're hearing about today means that we are shifting into a predictive and preventive model where, let's say, I can picture very clearly a world in which every person has their genome sequenced at birth, or maybe even before. If you do IVF, you can do this before. I told you that right now you can get DNA sequenced for about $200 if you just do the exome. We're predicting that within 5 years, it will be cheaper to flush a toilet. No, it will be cheaper to sequence your DNA than to flush a toilet. I was so excited, I got it backwards. I wanted to get to the punchline. The marginal costs of DNA sequencing are falling so rapidly that it's going to be pennies, maybe 2 US cents, in order to sequence a human genome by 2025 at the latest. So what that means now is that we have huge biological data sets. We have lots of ways of analyzing them and of extracting personalized real-time insights from this. And this isn't science fiction. This is happening right now. You probably saw that Google and Amazon recently entered the healthcare marketplace. And there's all kinds of questions around what that actually means. Like, who owns your DNA data? How many folks in here have done a genetic test, like 23andMe or something like that? A few. Okay. So who owns your data? When you do a 23andMe kit, who owns the DNA data? 23andMe owns it, right? So you have no way of knowing who they're selling that your DNA information onto in order to get money. And you don't know where that's going. And there's a whole industry now called bio brokers that are trying to flip that around so that you become the owner of your digital DNA information, and you're empowered to sell that on in ways that capture the value that your genome and your genetic information is adding to the larger environment. It's really, really exciting. So let's talk for a few minutes about agriculture, because I think agriculture is one of the most important applications of the life sciences. And I'm an American, so this is a little politically embarrassing, but I'm going to stand up here and say it while you're all looking at me. Climate change is real. Yeah, it's an existential threat to our planet. Thank you. Go science. It was caused by humans. And if humans caused it, that means we can fix it. Okay, whew, I got that out. All right, so if we're going to go on feeding an exponentially increasing population in the existential threat of climate change, we need to be using every single possible solution space we have available to us and every technology that we have. We need to increase crop yields. We need to increase nutrition. We need to figure out how can we grow crops in really salty soil because the ocean levels are rising. Or Jeff showed you that terrible picture of the drought. Those droughts are only going to get worse. So how do we get plants to be growing with a lot less water? We need to answer all of those questions right now. You guys probably saw last week there was a really alarming report that came out of an independent international body saying that not only are we like nowhere near hitting that 1.5 degrees C temperature restriction, we are like an order of, factor of 2 off from hitting it, right? And that's from countries that chose to join the Paris Agreement. Okay, that's the end of the politics, I promise. So let's talk about agriculture. Let's go back to this idea about debugging DNA, and let's talk a little bit about gene editing, because this is a really interesting innovation space. So this cow lives up the road from me in California at UC Davis. And this cow is special because it was born without horns. So normally when you're farming cattle, when the cow is young, it has to go through this process called debudding, where the farmers cut the horns out of the cow's head so that the cows don't fight each other. Turns out cows do all kinds of crazy things. I've been learning a lot about cows lately, including hitting each other with horns. So they always get rid of those horns so that you can stick more cows together in one place and they won't fight each other. It's unpleasant, it's expensive, it's miserable for the cow. But now with gene editing, it turns out you can erase the code for making horns from the cow genome. Cow never has the horns, never has to be debudded. Here's another example. These researchers were looking at the tomato genome. And what they did was they found 2 pieces of DNA that were about 50 base pairs apart, or 50 letters apart. And they realized that if you use gene editing to erase 12 of those DNA letters, then the tomatoes would flower sooner and would have, you could basically get through more growing cycles in a particular season, which means any given acre of land got its crop yield going up and can produce more tomatoes. And that's just a really, really simple example of how we could be using gene editing to do things like make photosynthesis more efficient. Getting light from the sun and converting it into calories that humans can access is actually a pretty shockingly inefficient system. We can do that better. And this technology is really exciting. Now, the other interesting thing is how it's kind of reframed the GMO debate. And I'll just say this flat out so that we're all on the same page. There is no credible evidence that genetically modified organisms negatively impact human health. It's just marketing. Okay, so that's really, really clear. So when this technology came out, people said, okay, well maybe this is a chance to finally get away from this whole quicksand about GMOs, because by definition, a genetically modified organism comes when you take the DNA sequence from one species and you stick it into another species. But I told you that CRISPR-Cas9 is like an eraser on a pencil. You're not adding anything new in, you're just changing what's already there. So this was this wonderful magazine cover, the cover of MIT Tech Review, where they were like, wow, OMG, maybe we can get rid of GMOs. The problem, though, is that different countries are regulating this differently. So in the United States, our courts came down and said, nope, GMOs and CRISPR does not give you GMOs, so that's fine. All of our regulatory framework doesn't apply to that. Europe gave the opposite rating. And so that was so disappointing to the scientific community, because it fundamentally not only misunderstands the way the technology works, but it's like taking a tool for future-proofing the world and just throwing it away. And that was really disturbing to me. So what that means for Africa is that, yes, it's going to impact your ability to sell crops that you grow through CRISPR-Cas9 and other gene editing in Europe. But what it also means is all those European competitors that you might have had to face, they can't innovate in this space. So there's a massive, massive opportunity for African innovation right here. It's really important, and it's a wonderful opportunity for South Africa's to be innovations in agriculture. And in fact, there are already some really exciting initiatives going on. There is a program in Nigeria called MoBreed, and another program called Genes that is looking at 10 priority underutilized crops that are underutilized globally, but are in the African ecosystem and in African traditional diets as being ones that you could innovate with, things like millet and sorghum and these types of things. So the goal, of course, is to increase yield and nutrition without making the environmental impact of agriculture, which is already really huge, even worse. So I think this is really, really exciting. So now let's think about information and data, because by definition, DNA is an information storage molecule. That is what it evolved on Earth to do. And what's really interesting about DNA as a chemical is that it's very, very stable for a very long time if you keep it cold and dry. You know, a DNA molecule will last 100,000 years as long as you keep it dry. Not true of your flash drive, by the way. So what does that mean? Well, what some people realize is that that means you could take DNA and use DNA as a way of getting at the major information storage crisis that we're having right now. So just to put that in perspective, if you were to take the entire internet, all of it, even the pornography, and convert that into DNA sequences, it would all fit in the back of one pickup truck. And all you had to do is keep it cold and dry, and it would last for tens of thousands, if not hundreds of thousands of years. So now what we can do is, okay, let's take a look at what it costs to make DNA if I'm going to use DNA as an information storage molecule. The blue curve is that same curve that I showed you earlier. And what you can see is that DNA synthesis, either making short pieces of DNA or making long pieces of DNA, is also becoming radically cheaper, but the curve you can see is much shallower. So you might say, all right, well, it's going to take a long time, Tiffany, for us to get to the point where DNA sequencing, we're reading DNA, is just as cheap as writing DNA. But the good thing is that Moore's law is a false analogy here. Because by definition, transistors can't make more transistors, right? That's what Moore's law describes. But DNA, by definition, makes DNA. So whenever you want another DNA copy, all you do, you don't have to go back and order another one. You just put it into an organism, and something alive will very happily copy it for you, because that's what life evolved to do. So we don't have to wait until 2040 to be seeing a revolution in how we can program life through information. And in fact, just a couple of months ago, a group already showed me that my curve was wrong, which always makes me happy. So we were already way below this on short DNA synthesis. And in fact, on Friday, which was so recent I couldn't even make a slide to show you, a new company in San Francisco called DNA Script shows that now they can build DNA 150 letters long, which was triple what we were able to do less than 6 months ago. So this is changing, and that's using an enzymatic method, a biological method, rather than a chemical method. Super important. This means DNA synthesis is about to go through a burst of getting faster and cheaper that's going to revolutionize how we use it. So this group, a group at Harvard, George Church's group, what they did was they used DNA to store not just static information, which people have done in a while. You can use a code like the one I put on the top of the screen here to convert zeros and ones from binary into DNA. That's actually really easy. But they figured out a really clever way to store a movie in bacteria as DNA, and then get the movie back out and turn it back into computer code. And what they did was, you can see the left side is the image, the movie that they started with, and the right side was the image that came out. It's not perfect, but you can tell what it is. And this is using E. coli, actually, as a computer, a repository for information. Super, super, really interesting. So what that means is that there are a whole bunch of companies out there that are making DNA synthesis faster and cheaper than ever before. And what this means is we're getting to the point where we're talking about drag and drop construction of new life. A group recently redid the whole yeast genome so that it becomes a chassis, like building a car, where you say, okay, I want to be able to eat oil and do photosynthesis. Add to cart, click, and then the Amazon drone delivers the DNA to you, and then boom, you're off to the races and you can start being a synthetic biologist. And so if we think of life as an operating system, and like an engineer would think of it, we can think of life as having inputs, and then something happens, usually with DNA, and then there's some kind of output, and then we can reach a goal. So that's being a synthetic biologist. It's treating life as a computer, and it's treating the biology as the technology. And so I want to challenge you to take a moonshot, like you've heard from several of my colleagues. And here's my challenge to you. Figure out how you're going to solve something using biology as the technology. Let me show you a super kind of silly example that I came up with to show you. So let's say you're like me and you're a scuba diver and you're a biologist and you really care a lot about the health of the ocean. Well, let's say I take a fish and I figure out a biochemical pathway in the fish, I make it, so that the fish can now eat plastic. That's already a win, right? And then it converts the plastic into some kind of alkaline molecule, and when the fish poops that out, it reverses ocean acidification, thereby saving the coral. It's insane, right? It's totally ridiculous. How could this possibly work? Well, that's details. But what I'm doing is taking a maker mindset and thinking about how I can use biology as technology to change one of the world's biggest problems. And there are hacker spaces all over the world where you can go, even if you're not a biologist, and try this stuff out with your own hands. My kid just turned 7 years old. He's done this a bunch of times. I mean, okay, he's 2 biologists as parents, so it's a little weird. But your kids can be doing it too, and you have biohacker spaces in South Africa. I looked them up. So go check it out and go be a maker and an innovator. DNA sequencing has done a lot of other things too, like shown us that our bodies are ecosystems. So you probably have heard of the microbiome, which is all the things that live on you and in you that aren't human, but are really, really important for your health. And I want to tell you, this is my particular area of expertise. So I just want to tell you about 2 things briefly that I think are why South Africa in particular should be interested in the microbiome. So this group did a study in India where they were looking at infant sepsis, which is the rate of newborns dying of overwhelming infections very, very soon after birth. Sepsis kills about 600,000 babies a year, and it kills older children and adults as well. So what this group did was they did the gold standard of scientific research, where they did a double-blind, placebo-controlled clinical trial. And what they did was they went to India and they found 5,000 infants who had just been born. And then what they did was they gave them what's called a symbiotic, which is a bacterium plus a sugar that the bacterium can eat. And they picked a bacterium that sticks around in the gut. And they gave each of those infants 1 dose for 1 week of this bacteria-sugar combination. And when they did that, they found the babies that got that dose had a 40% lower rate of sepsis than the other group who didn't get the dose. They had 80% lower rates of any kind of infection, a third less pneumonia, and absolutely no side effects. These results were so stunning that this study had to be stopped because it was deemed unethical to withhold this treatment from the other arm of the study. So let me ask you, how much money does it cost to save a baby's life? This way, what do you think? Yeah, $1 US. So when I think about the global health implications of what we can do with this type of information, it's just massive. I'm going to skip that, and let's go to the species level now, because there's so much going on here that's so interesting. We now have the power to impact life on a species level all over the world. Now, humans have been really good about doing this for like 10,000 years, right? We're really, really good at killing animals until they go extinct. But now we can actually do this on purpose. So what does that mean? So let's talk about mosquitoes for a minute. So, the burden of mosquito-borne disease is terrible, especially in Africa. So what if I told you we could genetically modify mosquitoes to never carry malaria ever again, to never carry Zika, to never carry Japanese encephalitis or dengue or yellow fever? We can do that. We can do that right now. It's not science fiction. And we can use this thing called a gene drive in order to force that new DNA through wild populations in a really clever way that you can also build a kill switch in. And the math indicates that within about 14 generations of mosquitoes, you could wipe out every mosquito on the face of the earth this way in a little more than 1 year. That's astonishing, right? And there are groups that are working on this right now in very targeted things to figure out, does this work? And what are the implications of being able to wipe out an entire species on purpose on a global level? So, and this isn't science fiction. A city in Australia has already done this, has already dropped their dengue rates to 0 levels. But this is super important, because if I can edit mosquito DNA, then I can edit human DNA too, right? There's nothing fundamentally special about human DNA. It's all the same thing. It's all molecules. So who gets to decide to do that? I think we can agree that using gene editing to treat cancer, that's a no-brainer, right? But what if I were to edit an embryo so that it would never get cancer? That's a little stickier, right? Because the embryo's not making the choice in order to get that treatment. And the question becomes even more important when we start thinking about non-therapeutic things. What if I wanted to make my kid a little bit smarter, or a little bit faster, or a little bit taller? Do I have the right to make that decision on behalf of the generations that are coming after me? And because these questions are so important, different countries and different ecosystems are treating this differently. China, it turned out, was quietly already doing this type of gene editing in humans and not telling anybody. When the day this news came out, I went and I looked online for what was going on in the US on the same day, and this was the headline. Yeah, we might be doing it any day now. Science is being balkanized because we have different frameworks that we're using. And in Canada, it is actually illegal. It is a felony for a scientist to edit the genome of something that could go on to be alive. I was giving this talk in front of a group of Canadian government people, and they were like, Dr. Vora, what can we do to foster innovation in Canada? I was like, get rid of this law. Don't make it illegal for your scientists to innovate. It doesn't make any sense. So I've been asking you a lot of big questions, and here's a tool I want you to think about as you're moving forward. I think about 4 questions. I think about, is there a need for this intervention or this technology? And that's like a global health kind of question that's pretty straightforward to answer. Can it be done? That's a technical question. I'm a scientist. I really like technical questions. Is it legal? That's a regulatory question and an ethical question. And should it be done? That's a moral question. And it is much, much harder to answer. And it's a question that we don't get to put off or make our kids answer. It's up to every single one of us in this room to do that. So here's my call to action for you. So you guys are the makers of the future. The future isn't something that's going to happen to you. You're going to design it. And by joining the community and meeting each other and doing all these amazing things, you are positioning yourselves to be the makers of the future. In September, your government committed to boost R&D spending from 0.8% of GDP to 1.5% of GDP in the next 10 years. Remember, your science budget has always been about the same as Norway's, and yet your population is 10 times larger than Norway's. But as your science minister recently pointed out, government can't do it alone. It's up to corporations, innovations, companies, and all of these other places to come up with the R&D that's going to be important. So this is where we started, right? I told you going back wasn't the answer. So my challenge to you is, how are you going to turn a cul-de-sac, a dead end, into a roundabout? What's your moonshot to make life better on this planet and maybe on another planet? And not only do I want you guys talking about that with each other during the breaks, but I'm going to be down there too. So come find me and come tell me how you're going to positively impact 1 billion lives on this planet using the life sciences. And that's where I'm going to stop. Thank you very much.